All right. Bill, what does the H stand for on the inactivation gate from your figure?
That's a terminology that comes from Hodgkin and Huxley, the famous people who first studied sodium channels using the voltage clamp technique. They wrote an equation that described the sodium current, the sodium signal, and they used two variables, M to control the activation process and H to control the inactivation process. I have no idea why they chose those letters, but they're just letters in a mathematical equation from the work of Hodgkin and Huxley, which is the classic in our field, really begins the modern era of sodium channel research in 1950s.
All right, great. Here's a question for Dr. Campbell. What is your hypothesis as to how the list variance, YS variance associated with the beta subunit leads to gain of function?
I think Professor Catterall suggested this during your presentation, that it's localizing, it helps, these beta subunits are help localizing to the proper location on the neurons.
They also help in the expression of the channel, by which I mean in the synthesis and insertion of the channel into the cell surface. The beta subunits, both beta 1 and beta 2, play a role in that process. So you end up getting more sodium channels if they are acting properly. Now, these mutations, which are gain of function, may mean that the beta subunit is even better at its chaperone function when those mutations are present, and you get more sodium channels, which would be a gain of function. It's also possible that those mutations are changing the interaction with beta subunits in some functional way, changing the gating.
All right, question for Dr. Sanders. Do we know what the percentage is of loss of function versus gain of function? And are those de facto two different problems which need different solutions?
Great. So the ratio is about 5 to 1, so 7.5 divided by 1.4. And the reason for that is it's much easier to break something than it is to make it do something special. It's throughout all genes, it's more common to see loss of function effects than gain of function. In terms of whether we need to do different things, so in a simplistic answer, the answer is yes. If it's gain of function, you want to reduce the amount of sodium coming in. If it's loss of function, you want to increase it. But when you start thinking about the mechanisms by which you might do that, I think it's really a place where the community comes together, and it really is a common disorder, because many of the tasks you would need to achieve to be able to make progress are common for both. For example, you would need if you're looking at gene therapy, you'd need a route of delivery and you'd need a mechanism, which would be the same, and you need to do extensive safety trials on both. The only thing which would differ would be the little bit which you put in the last bit, to which actually targets the gene therapy. For small molecules, it's probably a little bit more distinct, but still there, we still need to know the natural history. And of course, remember, it is just a model. It's not like everyone is exactly gain of function, exactly loss of function. It's really by understanding both that we'll come together and understand how this channel works.
Awesome. All right. There's so many great questions here. I'm just rifling through them all. Okay, great. One question was, does SCN2A turn into SCN8A with the loss of function group?
No, they're two different genes. So they sit there in the axon initial segment, and we don't exactly know why, but for some reason, SCN8A, which makes a protein called NAV1.6, displaces SCN2A. Maybe it's a bit of a bully or something. But I think that's one of the fundamental questions of sodium channels, is why what guides that distinction? That there must be some molecule which tells NAV1.6 where to go and where to bind. And we don't know what that signal is. But they're two different genes. SCN8A also can lead towards seizures and developmental delay.
Great. Here's a question for the team. What are the long-term repercussions for prolonged blockage of the sodium channel with sodium channel blocking medication?
Is that for me?
Counted to the T.
So, well, I mean, the first one is if you block all the sodium channels, the brain stops and the heart stops. It's a bad side effect. Those drugs can be used in therapy for the reason that I mentioned on my very last slide, or last few slides. They bind within the pore of the channel. They bind there better when the channel opens. And so in a disease where you have hyperexcitability and many action potentials being generated, the channel opens more often and the drugs enter and block more effectively. And this is called use-dependent block. So if the channel is used, it gets blocked better. And so anti-epileptic drugs work in this way to some degree. Anti-arrhythmic drugs, as you might guess, when the heart beats fast, the sodium channels open each time, there's an extra heartbeat, and so those drugs act in the same way in the heart. And in fact, the local anesthetics, which I use as an example in dental pain, again, the pain signals are generating action potentials at high rates, just like you saw for the horseshoe crab eye with a lot of stimulus with light. If you have a lot of stimulus with pain, then your nerves are generating a lot of action potentials. Those local anesthetics block the nerves that are firing rapidly more effectively than the neighboring nerves. So you don't feel the pain, but you can still tell you've got a lip there because the other nerves aren't blocked as much. So that's a crucial aspect of these drugs. And the existing drugs in the clinic block all sodium channels. And so that's a big problem because if you don't want to solve an SCN2A, mutation by blocking SCN1A, 2A, 6A, and every other, there are 10 sodium channel genes. So the side effects would be terrible. So that's why the emphasis in drug companies is on sodium channel blockers that would be, as we say, subtype selective. They would hit SCN2A and not anything else, and so on.
I just add, this is going to be an important theme we see throughout this, that there's firstly this challenge of trying to hit SCN2A rather than 8A or 1A or 4A, many of which have other effects. And they are very, very similar to each other. The second challenge is it's a bit like Goldilocks. You don't want too much, you don't want too little, you want it to be just right. And that's much harder than just replacing a missing gene. We need to have a therapy which tailors it to the right amount in the right context.
Just staying on the medication theme, question is, what medications work best to manage sodium channel gains versus losses in general?
Well, I'll start, but you should finish up. So the existing medications are all inhibitors. And so they're really only useful, I think, in a gain of function setting. And so they probably would be useful for SCN2A patients who have epilepsy. And they probably wouldn't be useful for SCN2A patients who have developmental delay and autism. And really, Kevin's beautiful work has made this dividing line. And I think that's a generally correct statement.
Yeah, so for the gain of function, you want medications which block sodium channels, so anti-epileptics which block sodium channels. In terms of the specific ones, I come from a pediatric background and just look them up when I need them, but I'm sure there's people in this room who would be much, much better at answering that. For the loss of functions, there's some children with autism or developmental delay also get seizures, and in that case, you want to not block the sodium channels, which makes sense when you think that there's less sodium there in the first place. We had wondered early on if the seizures in autism might be a reaction to development with too little autism, and you essentially overshoot. But that doesn't seem to be the case because the sodium blocking channels drugs don't make it better. At the moment, there are no medications for developmental delay or autism in any context, and our hope is that SCN2A leads us towards something like that. But that's really is cutting edge. When we try and divide the data up by into the two groups, it's the age of onset of seizures which we use to make the distinction. Following your point that both groups can have developmental delay.
Great. One more medication question, then we'll move to a different theme. This is a question addressed to Dr. Catterall. Why does lidocaine treatment, which was used with my daughter and gave her a month of 0 seizures, why did that lose effectivity over time? It was a short miracle for her at 6 months of age, and then it became less and less effective until it no longer did anything regarding her seizures.
I'm sorry, I don't have a good answer for that one. Usually lidocaine is not given for treatment of seizures. Usually Dilantin, for example, or lamotrigine, other sodium channel blocking drugs are used for treatment of seizures. And lidocaine is given generally, that is not by just injection in a specific site as in the dental case, but it's given generally for treatment of cardiac arrhythmias, where it's one of the most common antiarrhythmic drugs. It's used in a clinical, in an urgent care setting in the operating room and so on. So I'm first surprised that there was an effect, and unfortunately, I don't really have a good idea what might have changed it, although as you've heard, the sodium channels that are being used by the brain to generate action potentials change over time. Perhaps there was, that was part of the basis for the changes that were observed in the drug effectiveness.
Great, great. A question for Dr. Campbell. In your slide listing all the letters representing the SCN2A gene, can you just explain again what all those letters mean?
So,
each and every one.
So those letters represent the amino acids that code for NAV 1.2, so the protein structure. So those DNA encodes for, is transcribed to RNA, which then is transcribed to the amino acids, which build the protein structure.
All right. Question here is, how do we discover where our specific mutation occurs?
How do we what?
How do we discover where?
So we built a little tool where you can put your mutation in, and it plots it onto onto the gene. And I think it's on the SCN2A family's website. Yeah. Yeah. Fantastic. So you can put in 2 things. You can put in actually 3 things. You can put in the DNA coordinates, the cDNA, which starts with a C, or the protein, which starts with a P. And so the protein's probably the easiest one to do. And you'll see a capital letter, a number, and another capital letter. And the way to translate that is there's 22 different amino acids. If you think of like sort of Lego building blocks, like there's 22 different colors. The first capital letter is the normal amino acid in that position. The number is the position, so it's the number out of 2005, 1 being the first, 2005 being the last. And then the last letter is the change. So you might see an R853Q is an R, arginine, at 853 changing to a Q.
So my postdoctoral fellow, Samaya Akbal, she's coming out with a paper that has a website called Miscast for the protein structure part. So people will soon be able to just type in the protein, the amino acid that they're interested in, and see where it is on the protein structure.
All right. All right. Here's a question addressed to the panel. What do the beta subunits do?
So we think of them as being auxiliary subunits, that is, and not directly involved with voltage gating or opening the pore or so on, as I mentioned. So when the beta subunits assemble with the alpha subunit, that complex is more likely to go to the cell surface and be functional. So they help in that way. You could think of that as like being a chaperone, you know, taking the date to the dance, taking the alpha subunit to the cell surface. They also modify the function of the alpha subunit. That is to say, with the presence of the beta 1 subunit, some sodium channels, SCN2A as an example, gate at more negative membrane potential, so they open more easily if the beta 1 subunit is associated with them. So they have modulatory effects on channel function, and they have what you might think of as cell biology effects, helping the channel to get to the cell surface and also to get to the right place. As Stephan showed nicely in his slides, sodium channels are at axon initial segments. They're not everywhere. They're at high concentration at axon initial segments. And so interactions not only with beta subunits, but with components of the cytoskeleton, the protein skeleton inside the cell, helps to get the channels to the right place and to hold them there. So they have very important functions, but they're not the pore or the voltage sensors or those integral parts of channels.
All right, question for Dr. Sanders. Yeah, you talked a little bit about estimated prevalence in your talk. Can you break that down by each of the clinical presentations?
Sure. So in 2017, after the last family group, a load of us worked together on a review, including some families here and many of the scientists in the room. In that review, there's a table which spells out exactly how we derive these estimates. But broadly, that 1 in 10,000, it's 5 to 1, 5 loss of function to 1 gain of function, which means roughly 5 autism developmental delay to 1 infantile epilepsy. Now, of course, when you look in the room here, that ratio is not the case. I expect it's probably more like 1 to 1. The reason for that being, if your child has a seizure in the first 3 months, you're almost guaranteed to get genetic testing. If your child has autism, unless you're at a really, really good teaching hospital with a very active genetics program, it's unlikely that child's going to get tested. So we're not very good at picking up the loss of function ones. That table spells out exactly how we got the estimate, but it's 1.4 in 100,000 for infantile epilepsy and 7.5 in 100,000 for autism, which means that roughly 0.3% of cases of autism are due to SCN2A.
Wonderful. That was great. Thank you. So we've got just about 2 seconds to say a quick thank you to our speakers here today. Leah's going to bring up a couple of little things here as well. But thank you to the 3 of you. Since I get the microphone, I can say this right now. Thank you to 3 of you. That was incredibly informative. A lot of great science. The work you guys are doing is amazing for me to sort of watch and see and learn from. So thank you very much. A quick round of applause. We'll take a break.